Hello, everyone. My name is Alka Chaube, and I am the Chief Medical Officer of Bionano Genomics. And today I will be talking about next-generation cytogenomics with optical genome mapping. And this technology is now here and to stay. So before I begin, let me just walk everybody through the timeline of the evolution of cytogenetics to next-generation cytogenomics and where we are today. If you see that when the term chromosome was coined in 1888, it took a significant amount of time until 1970s when chromosome banding techniques were developed. The evolution from chromosome banding to fish then also took several decades. And then these technologies became the standard of care for constitutional genetic testing and disorders, and also for hem-onc applications later on. But then it wasn't until the early 2000s when microarray came into the clinical space, and various medical associations recommended replacing karyotyping with chromosomal microarrays for them to become first-year testing. Then sequencing technologies evolved during that same time frame. And now we have next generation sequencing panels, exome sequencing and genome sequencing also in the clinical realm. So where are we today? Now we have entered the clinical field with optical genome mapping. And today we will see the potential of optical genome mapping in both constitutional genetic disorders and neoplastic disorders. So what is optical genome mapping? Optical genome mapping is this beautiful technology where we are able to visualize and map these enormously long molecules of DNA, several hundred kilobases to megabase length molecules that are moving through these nanochannels on the sapphire chip, which you see in this lower image over here. These molecules, after the processing and after labeling, are linearized in these nanochannels and then are imaged in our instrument, the sapphire instrument. What you see in this image here is these long molecules that are labeled with a specific motif, 6-nucleotide motif and a fluorophore to allow for pattern recognition for evaluating the structural variations in the human genome. So this is a slide courtesy Dr. Alex Heushen from Radboud University Medical Center, where we see the chromosome banding pattern, and each band can be from 5 to 10 megabases in size. And this is the technology that is used in routine cytogenetic labs across the world. What we see by optical genome mapping now is, again, these same long molecules of DNA and this 6-nucleotide specific fluorophore that you see in green. These motifs are also giving us patterns for recognition of structural variations. So this is a simplified workflow of how optical genome mapping works. So the first step is to isolate very high molecular weight DNA. And this is very important for getting these long molecules for accurate structural variation detection. The next step is labeling the specific sequences across the entire genome. And this is done using a specific enzyme. These labeled molecules are then transferred onto the sapphire chip for scanning. And then, as I showed in the video earlier, those molecules moving through those nanochannels are imaged. And then using specific algorithms, the genome and those molecules are assembled into consensus maps and then compared with a reference to detect structural variations. There is another algorithm that is used to align the molecules to the reference and counted to measure dosage. So this is similar to how laboratories are used to looking at a microarray profile to identify whole chromosome gains and losses. And these structural variation maps are used to look for different classes of SVs. This is an example of a deletion where you see a reference map and then the sample map of interest. And I'll go into details in the next slide. So structural variants are essentially called by comparing maps to a reference or to each other. So what we see in the top in the green is the reference map with these specific patterns. And the assembled maps are in blue from a given clinical sample to identify the different structural variations that might be occurring. So what you see in the left-hand side here is a deletion, which can result from the loss of labels or a decrease in the distance between two labels. Similarly, an insertion can be due to the introduction of new labels or increase in distance between two labels. Copy number aberrations such as repeat array expansions can be very easily quantified by just counting the labels and the distance between the labels. And changes or aberrations such as duplications can be very easily mapped, and not only mapped, but their orientation can also be very easily deciphered by just looking at the pattern recognition of these labels. Other unique classes of SV, such as translocations, can be easily identified where two different chromosome reference maps, the pattern on those can be used to see where the translocation involving two or multiple chromosomes is taking place. Inversion is also an example where just looking at the pattern of the labels and the distance between the labels, the inversions can be very easily detected. And as I mentioned on the previous slide, there is a unique algorithm to also look at the copy number variation profile of by optical genome mapping. This is a table showing different structural variation classes that are detected by optical genome mapping. And we will start with whole chromosome changes. Monosomies and trisomies can be easily detected. And as I just mentioned, copy number variations such as deletions, whether they are terminal or interstitial, or duplications, whether they are tandem or inverted, or inserted into another part of the genome, can be very easily detected. Insertions, whether they are of unknown or known sequence, can also be accurately identified. And translocations, which are very unique and very difficult to identify using molecular methods, can be very easily detected by optical genome mapping, whether they are balanced, unbalanced, or cryptic. Inversions, whether they are paracentric or pericentric inversions, just based on the fusion pattern of these structural variation maps, can be identified. And other unique structural variations such as ring chromosomes can also be very easily detected. Currently, we are working on improving our algorithm to pick up loss of heterozygosity or absence of heterozygosity, but macrosatellite and microsatellite repeat regions of the genome that lead to specific disorders such as facioscapulohumeral muscular dystrophy 1, which is a repeat contraction disorder, and Fragile X, which is a repeat expansion disorder, can be easily detected. We are not a sequencing company, so we do not interrogate or detect single nucleotide variants. Bionano Sapphire system for optical genome mapping provides a complete workflow for genome analysis. Starting from sample preparation, there are specific labeling kits, specific DNA isolation kits. There is a unique Sapphire chip with the nanochannels, as I showed you on the previous slide, and the instrument itself where all the imaging of those molecules happen. But not only that, Bionano also provides the bioinformatics solution and the software, which is called the Access software for visualization and analysis of these structural variants. So now let's focus on the unique applications for constitutional genetic disorders for next generation cytogenomic tool that optical genome mapping can be used for. If we look at what are the capabilities of the tools that are in current use in a classical cytogenetic or cytogenomic lab, there are three main technologies: karyotyping, fluorescence in situ hybridization or FISH, and chromosomal microarray or CMA. If we look at the variant classes and detection, what you see here are the limit of detection of each of these technologies, such as karyotyping, FISH, and microarray. Compared to these three technologies, it is very clear that optical genome mapping can pick up the majority of the structural variation classes, except single nucleotide variants, which I have alluded to earlier also. So now let's look at some of the examples of different classes of structural variations that are important in diagnosing a specific disease. So microdeletion syndromes and microduplication syndromes are one of the most common classes of structural variations. And here is an example of a microdeletion syndrome of chromosome 22q, which is clinically known as the DiGeorge syndrome. And this syndrome can be of a varying size from approximately 2.8 megabases to even a nested deletion syndrome, resulting into a specific phenotype. What you see in the slide is the copy number track, which is also showing the deletion. Along with these, our structural variation maps, our consensus maps, which are also showing the large deletion of 2.8 megabase in size. What is really intriguing here is that there is an inversion that is occurring on the opposite strand, which can very easily be seen by these consensus maps and the molecules on the other allele. This kind of information is missed by most of the classical methods, and this is where optical genome mapping has the power to provide this information, and this could be one of the reasons that would facilitate a deletion taking place. Our next example is an introgenic deletion, where everybody knows that Duchenne muscular dystrophy phenotype results from single or multiple exon deletions of the DMD gene or duplications, and a small fraction of DMD patients result from sequencing variants. What you see is when you open the Bionano Access software, the first thing that will be seen is the circus plot where the entire genome is viewed. And this is from chromosomes 1 through 22 and X and Y, which is the outermost ring is the ideogram. This is followed by the consensus gene list or any BET file that the users might want to use for interrogation of a clinical sample. The next ring from the outside is showing the different classes of structural variations with different colors. So you can see here insertion, deletion, inversion, duplication, and translocations. The next ring from the outside is showing the copy number variation track, where large deletions, duplications, or aneuploidies can also be visualized. The innermost ring will show whether there is an inversion or a translocation or multiple translocations or chromotryptic events that might be happening in the genome. So in this example, what you see here is that there is a deletion on chromosome X, and if you hover over that and when you click on it, then the maps will usually open. Again, you see the chromosome X reference pattern in the top, and then the sample of interest, where this is showing a single exon 45 deletion of the DMD gene, approximately 165 kilobases in size. Our next class, which is very important, is the ability to pick up repeat expansion disorders. And this is a classic example of Fragile X syndrome. In constitutional disorders in children with intellectual disability or developmental delay, microarray and Fragile X testing is the first-year testing as the standard of care. And what you see here is that this results from a repeat expansion of CGG repeats in the promoter of the FMR1 gene, And what we see here is an insertion of approximately 866 base pairs in the promoter region, which is consistent with more than 200 repeats in the FMR1 gene. These are results from a well-established Fragile X cell line, and this insertion is completely novel to our Bionano control sample structural variation database. Another unique feature that optical genome mapping allows for the diagnosis and detection is the ability to pick up repeat contraction disorders. This is an example of FSHD1. This is a very complex genetic disorder, neuromuscular disorder, which results from a repeat contraction of these D4Z4 repeats on chromosome 4. This is complex because the same cluster of D4Z4 repeats also occur on chromosome 10. And by using sequencing technologies and other conventional technologies, it is very difficult to discern between chromosome 4 and chromosome 10. However, because optical genome mapping allows the users to interrogate the patterns of these labels on these ultra long molecules of DNA, it is very easy to identify not only the repeat contraction in the top portion over here of a contracted allele compared to a normal allele, but also gives the pattern for the permissive allele, which is the 4qa polymorphism, which is approximately 10 megabases downstream of the D4Z4 cluster. And this is the pathogenic allele which results into the disorder. So using optical genome mapping, this is so easily discerned and can very easily be counted. The repeat contractions can be counted just by comparing the distance between the labels. To enable the customers to quickly get to a report for their clinical reporting as part of their lab developed test, we also offer an nFocus FSHD analysis tool, which is a very quick analysis, which requires less than 2 hours to get to a report, which will accurately give the different haplotypes, the alleles, whether it is 4qa, 4qb, what are the counts of the repeats, whether they are 3 count or 28 count, and also give the information of what is happening on chromosome 10 just to differentiate this disorder. Let's switch gears and move on to how optical genome mapping can be used for next generation cytogenomic applications, especially for heme malignancies and for other solid tumor applications. This is an example of translocation involving oncogenes. And what again you see at the top is the circus plot showing the 1114 translocation over here along with another translocation. If you zoom in onto the 1114 translocation, in B, what we see is the circus plot just showing 2 chromosomes, chromosomes 11 and 14. And for those of you that know about this, this is the classic 1114 translocation involving IGH, CC, and D1. You can see from the SV maps at the top, we have chromosome 11 reference in green. At the bottom, we have chromosome 14 reference in green. And in the middle is the blue map, which is showing the alignment of these different patterns to chromosome 11 and 14, and also giving as close as possible the labels which are disrupting these genes, which are going to give the information about where the breakpoint of these translocations are taking place. Another structural variant very important for chronic lymphocytic leukemia is deletion of 6q. And here the copy number variation profile can be used to see that entire arm, long arm of chromosome 6 is deleted in this sample, along with other aberrations also taking place. Similarly, one of the classic examples for acute myelogenous leukemia is the translocation involving chromosome 8 and 21. Again, in the circus plot, we see the 821 translocation along with a few other translocations taking place. And when you zoom in, you can see that on chromosome 8 is the RUNX1T1 gene, and then 21 is the RUNX1 gene, which is the hallmark translocation of AML. This is how conventionally 821 translocation is identified in bone marrows of these leukemia patients. And you can see that the bone marrow morphology can vary dependent on the clinical sample. And then the fluorescence in situ hybridization panels are used to try and identify if any translocations are taking place. If we look at guidelines such as NCCN or National Comprehensive Cancer Network, or the guidelines set by World Health Organization, what we see is that for most of these hematological malignancies, certain panel or certain translocations or structural variations are considered hallmark to try and establish favorable, unfavorable, or intermediate risk for patients with leukemias and lymphomas. If we try and interrogate and see, well, in one assay, how would a virtual AML panel with optical genome mapping look like? This is an example of a few of the abnormalities as per the recommendations, and you can see a deletion of 5q involving the CSF1R gene, which is again a 5q deletion is one of the hallmarks for AML, along with chromosome 7 or 7q deletions, which usually by FISH is just tested using the centromeric probe. And then any translocations or aberrations or amplifications involving the KMT2A gene on chromosome 11q23. And as I just showed previously, translocation involving chromosomes 8 and 21, and then the inversion 16 or translocation 1616 resulting into CPFB and MYH11 rearrangements can very easily be interrogated in one assay. So this heme malignancy panel, depending upon the leukemia or the lymphoma, or what different structural variations we are looking for, can be very easily customized in the Access software. This is some of the studies that have been recently published using optical genome mapping data. This is a study which was performed in the United States by multiple centers using optical genome mapping for the assessment of genomic aberrations in AML. And this study identified 100% concordance in 100 AML samples. As you can see, this was a study done at Columbia University with collaborators from Mayo Clinic, Penn State, Path Group, Legacy, Augusta University, Fred Hutchinson Cancer Center, and MD Anderson. What this study did essentially was take 100 AML samples, which were analyzed in conventional cytogenetic labs by karyotyping. Some of these samples had FISH and or microarray done, and optical genome mapping using Sapphire detected 100% of the clinically relevant structural variations and CNVs in all of these samples. The most interesting thing here was that additional new information to approximately 11% was also identified by this study, which results into an increase in diagnostic yield of approximately 6 or 7% in cases that had a negative or a normal karyotype. So this is really important, and one of the recommendations that came from this publication was that optical genome mapping be considered as the first line test for the detection of clinically relevant structural variations in AML. Another study on constitutional samples done by Dr. Leila El-Khattabi's lab in Europe also showed 100% concordance in 85 samples that had constitutional disorders. Again, this was compared with previous data by karyotyping fish or microarray, and aberrations were detected by either the copy number variation tool or the structural variation tool, as I've just showed you. And these are the classes of the aberrations, and you can see all of them were picked up, whether deletions, duplications, translocations, inversions, aneuploidies, or even ring chromosome. This study by Dr. Shi et al. From Dr. Pui Kwok's group is extremely interesting because this was done on a subset of cases that were negative by microarray and exome sequencing. And what this study found was that using Bionano or optical genome mapping, there was an additional increase in diagnostic yield to up to 18% that was identified in these individuals that had a rare constitutional genetic disorder. So this publication was on 50 patients with congenital anomalies, as I just mentioned to you. And they used optical genome mapping along with the 10x Genomics linked reads. And there were 18% of cases where an additional diagnosis could be made from just using optical genome mapping alone. And this included a de novo duplication, but as you can see, not only identifying that it is a copy number gain or a duplication, but also the orientation of the duplication was very easily deciphered using optical genome mapping. And de novo translocations were identified in a proband with neuroblastoma and developmental delay. So now if we switch gears again, and now let's compare where does optical genome mapping compare to when we put sequencing technologies for copy number variation detection. So this is just an extension of the table that I had shown during one of the earlier slides. And now what this adds is, where does sequencing or next generation sequencing and the evolution of exome and genomes fall into place? What you see here in terms of all the different structural variation classes, sequencing, it only will pick up the single nucleotide variants, which optical genome mapping does not interrogate at all. However, translocations, inversions, repeat expansions, repeat contraction disorders are not that amenable to sequencing technologies. And this is where optical genome mapping has an enormous strength. This is an example of a female with a reproductive disorder. And what we see here is a balanced translocation. And unless there is a priori information for sequencing technologies, or there are unique capture methods, balanced translocations are not easily detectable by whole genome sequencing. But what we see here is the translocation involving chromosomes 7 and 10, and this is probably the reason why this individual had a reproductive disorder. This is the example of where, when you click and when you try to zoom in, are there genes of interest that could be playing a role? But you see here that on this structural variation map, which is mapping to chromosome 7 and chromosome 10, we see that the translocation breakpoint is not within a gene, but close to certain genes of interest. And this can, again, be very easily detected by optical genome mapping. You can see the reciprocal breakpoint also, where these maps are aligning to the two different chromosomes, 7 and 10. One of the most important things about optical genome mapping and its comparison with other technologies can be highlighted from this publication. So 2 large consortium studies, which have been published in Nature Communications, have utilized optical genome mapping, where optical genome mapping detected structural variations missed by next generation sequencing. So this is part of the 1,000 Genomes project, and where Bionano identified more than 8.5 times insertions and 35% more deletions than sequencing. So this has been published. This is data that is available for the viewers. So there is a December 2020 update on the data and the samples from this consortium, where the Human Genome Structural Variation Consortium also found that long-read sequencing technology of PacBio could only detect 72% of the large SVs detected by Bionano or optical genome mapping technology. So this itself, when you look at all the different classes of insertions to deletions, is that the Human Genome Structural Variation Consortium found that Bionano detected more structural variations greater than 1 kilobases in size than next generation sequencing or long read sequencing combined. So this really highlights the power and the potential of optical genome mapping in identifying all the different types of large structural variations that are happening in the human genome. So to conclude is how I started, that optical genome mapping is a next generation cytogenomics tool. As you have seen in both constitutional genetic disorders and hematological malignancies and other solid tumors, this technology is now here. And there are a number of users that have adopted this as a lab developed test for clinical use, as is every technology in clinical practice right now today. So optical genome mapping allows for the detection of all classes of structural variations occurring in the human genome. And I hope that you've seen that since different technologies are employed in tiered testing as part of a clinical workup, optical genome mapping also removes the need for tiered testing, just by its ability to provide all the answers for all types of structural variations. In constitutional disorders and for those applications, optical genome mapping can not only identify all copy number variations of clinical significance, but also will pick up all the balanced translocations and inversion events, along with the ability to identify repeat expansion disorders and repeat contraction disorders. For hem-onc applications, as you have seen on few of the cases that I showed today, optical genome mapping can pick up all actionable structural variations as per the NCCN WHO guidelines, which can be very easily implemented into a lab. Not only that, optical genome mapping does not require a huge bioinformatics core or tools that are usually needed for sequencing technologies. Our Bionano Access software solution is really easy to use and allows a graphical user interface for filtering and manual evaluation of each structural variant of interest for the lab and for the clinical sample. Not only that, from sample to answer, from DNA extraction to visualization in Bionano Access can be done in as little as 4 days. This really demonstrates the fast turnaround time that is needed for some of these clinical disorders. Multiple studies have demonstrated 100% concordance with conventional cytogenetic or cytogenomic methods, including karyotyping microarray FISH for both constitutional and hematological malignancies. And a recent study in the United States by UCSF has shown an increase in diagnostic yield using optical genome mapping in rare genetic disorders. So I do need to acknowledge all of our customers, and the data that I have shown are from their laboratories. So Dr. Alex Huyshen from the Netherlands, Dr. Blanca Espinet from Spain, Dr. Jim Broach from the United States, Dr. Bryn Levy from the United States, and Dr. Lela El Khatabi from France. And of course, I do need to thank Bionano Genomics, all the efforts that are going on in assay development, product development, and Dr. Alex Hasty for helping me put this presentation together. Thank you.